the 6 chromosomal basis of Mendelism. Here are the contents. What causes organisms to develop as males or females? Why are there only 2 sexual phenotypes? Is the sex of an organism determined by its genes? So this is a male and female Drosophila. The male has white eyes because it carries a mutation on its X chromosome. Chromosomes. Each species has a characteristic set of chromosomes. Within it you have chromatin, euchromatin and heterochromatin. Chromatin is a complex of DNA and proteins in eukaryotic chromosomes, originally named because of the readiness of which it stains with certain dyes. Euchromatin, it is the one that is not stained so intensely by certain dyes during interphase that comprises with many different kind of genes. Heterochromatin, they are the one is stained darkly even during interphase, often containing repetitive DNA with a few genes. So you can see here in this picture, can you spot which one is the euchromatin, heterochromatin and regular chromatin? So as you can see on the bottom left is the nucleus. This nucleus, there are parts which are white, so they are euchromatin, they are not stained. But heterochromatin, the other one which is dark. You can see the nucleolus in the middle. Euchromatin are not stained and the heterochromatin are the one which are dark. Chromosome number. Within a species, the number of the chromosomes is almost always an even multiple of a basic number. In human beings, for example, the basic number is 23. Mature eggs and sperm have this number of chromosomes. Most of the type of human cells have twice as many, 46, although a few kinds such as liver cells have 4 times the basic number, which is 92. The haploid or basic chromosome number and defines a set of chromosomes called haploid genome. Most somatic cells contain 2 of each chromosomes in this set and therefore diploid, 2n. Cells with 4 of each chromosome are tetraploid, 4n. Those with 8 of each are called octoploid, 8n, and so on. Basic number of chromosomes varies among species. Chromosome number is unrelated to the size of biological complexity of an organism, which most species containing between 10 to 40 chromosomes in each genome. The muntjac or tiny Asian deer has only 3 pairs of chromosomes on its genome, whereas some species of ferns have many hundreds. The record for minimum number of chromosomes belongs to the subspecies of the ant, the Mesia pedosula, in which the females have a single pair of chromosomes. Species reproduced by a process called haplodiploidy, in which fertilized egg diploid become females, while unfertilized egg haploid develop into males. Hence, the males of this group of ants have in each of their cells a single chromosome. The record of maximum number of chromosomes is found in the fern family Ophioglossum reticulatum. They have 630 pairs of chromosomes, which are 1,260 chromosomes per cell. It is remarkable that they are able to accurately segregate this enormous number of chromosomes during mitosis. So this is the chromosome number in different organisms. Simple eukaryotes, you have yeast with 16 haploid chromosomes, bread mold have 7, green algae have 17. For plants, for maize you have 10, red wheat is 21, tomatoes 12, broad bean is just 6. And then you have giant sequoia, is the largest tree in the world, 11. Crucifer is just 5. In invertebrate animals, fruit fly have 4, mosquito have 3, starfish have 18, nematode have 6, mussel have 14. Human being, we have 33 haploid chromosome number. Chimpanzee have 1 more than us, 34. Cat, they have even more chromosomes than us, 36 haploid chromosomes. Mouse have 20. Chicken has 39, even more than us or cats or chimpanzees. Toad 17. Fish 25. Sex chromosome. In some species such as grasshoppers, females have 1 more chromosome than the males called the X chromosome. Females of the species have 2 X chromosomes and males have only 1, XO. During the meiosis in the male, the solitary X chromosome moved independently of all the other chromosomes and is incorporated into half of the sperm. The other received no X chromosome, so become XO. In many other animals, including human beings, males and females have the same number of chromosomes. Y is much shorter than the X. If fertilization were to occur randomly, approximately half of the zygote would be XX and the other half would be XY, leading to 1:1 sex ratio at conception. However, in human beings, Y-bearing sperm have a fertilization advantage, and the zygotic sex ratio is about 1:3:1. During development, the excess of males is diminished by differential viability of XX and XY embryos, and at birth, males are only slightly more numerous than females, sex ratio of 1:07:1. By the age of reproduction, the excess of males is essentially eliminated, and the sex ratio is very close to 1:1. Human X and Y chromosomes. Terminal regions are common in both sex chromosomes. The X and Y chromosomes are called sex chromosomes. All the other are called autosomes. So the chromosome theory of heredity. By 1910, many biologists suspect that genes were situated on chromosomes, but they did not have definite proof. Thomas and Morgan discovered an eye color mutation in fruit fly. So fruit flies reproduce quickly, prolifically, and was inexpensive to rear in the laboratory. Plus, they have only 4 pairs of chromosomes. The X and Y chromosomes were morphologically distinguishable from each other and from each of the autosomes. So if you look at the picture there, those are the chromosomes that is in the fruit fly. Can you determine which one is the X and Y? So you can see there on the left, on the bottom 2, that's the XX. For the male on the right, you can see on the bottom you have XY. The Y is the one that looks like a V there. Okay, so you know at first glance the male is the right, the female is the left. So Morgan's experiments commenced with his discovery of a mutant male fly that had white eyes instead of the red eyes of the wild-type flies. When these males were crossed to wild-type females, all progeny had red eyes, indicating that white was recessive to red. But when this progeny was intercrossed with each other, Morgan observed a strange segregation pattern. Transmission of the mutant condition in association of sex suggested that the gene for eye color was present on the X chromosome but not on the Y chromosome. Because in this crossing, the females are all red-eyed. There is no white-eyed female. Okay, but for the red-eyed female, one of them are heterozygous. So meaning that the males usually will have the white eye mutation compared to females. So this is a case called hemizygote, an organism that only has one copy of a gene. Morgan carried out additional experiments to confirm the elements of his hypothesis. So he crosses between a heterozygous female and a homozygous mutant male. He crossed white-eyed females with red-eyed males. This time, all of daughters had red eyes and all of sons had white eyes. When he intercrossed this progeny, Morgan observed the expected segregation. Half the progeny of each sex has white eyes and the other half has red eyes. Chromosomes as an array of genes. Morgan and his students soon identified the other X-linked gene in DROSOPHILA. In each case, simple breeding experiment demonstrated that recessive mutations of these genes were transmitted along the X chromosome. Morgan research group also identified genes which were not on the X chromosome. So these genes followed the Mendelian principle of segregation, but they did not segregate with sex, as the gene of eye color did. So Morgan's students were able to show that genes were indeed situated at different sites or loci, from the Latin word for locus, which is singular, on linear structure. All genes were located on chromosomes, and Mendel's principle could be explained by the transmissional properties of chromosomes during reproduction. The idea, called the chromosome theory of heredity, is the unifying theory stating that inheritance patterns may be generally explained by assuming that genes are located in specific sites on chromosomes. Non-disjunction as proof of the chromosome theory. Bridges performed one of Morgan's experiments on a larger scale. He crossed white-eyed female DROSOPHILA to red-eyed males and examined many F1 progeny. Although as expected, nearly all F1 flies were either red-eyed females or white-eyed males, Bridges found a few exceptional flies, white-eyed females and red-eyed males. White-eyed females and red-eyed males. The exceptional males all proved to be sterile. However, the exceptional females were fertile. Bridges explained these results by proposing that the exceptional F1 flies were a result of abnormal chromosome behavior during meiosis in the females of the P generation. Ordinarily, the X chromosome in this female should disjoin or separate from each other during meiosis. Occasionally, however, they might fail to separate, producing an egg with two X chromosomes or an egg with no X chromosome at all. Meaning that, if you look at the picture on the right, look at the one below. Supposedly, you have one copy of each chromosome, but in this case, in non-disjunction, both of the pair go to the same cell. Non-disjunction. It is failure of disjunction or separation of homologous chromosome in mitosis or meiosis, resulting in too many chromosomes in some daughter cells and too few in others. Examples: in meiosis, both members might pair together and go to one pole instead of separating into two poles. In mitosis, both sister chromatids can go to the same pole, leaving none for one pole but two on another. So Mendel established two principles of genetic transmission. So allele of genes segregate from each other, and allele of different genes assort independently. Finding that genes are located on chromosomes made it possible to explain these principles as well as exception to them in terms of genetic behavior of chromosomes. Normally, in the principle of segregation, you know that each one of the chromosomes will segregate into different daughter cells. So you have maternally inherited chromosome with big A, big A. Paternally, you have small a, small a. So they pair together in the equator. In anaphase, they disjoin. Big A and small a segregate. Telophase 1, each chromosome would separate into a different cell. Big A in one cell, small a in another cell. In telophase 2, each allele is recovered in half of the product of meiosis. So instead of having big A, now you have big A only in one cell, and then another big A in one cell. Same goes with small a, small a. It now become small a one cell, small a another cell. So in independent assortment, two distinct metaphase alignment of maternal and paternal chromosomes are possible. You have big A, small b, and small a, big B. Okay. So during metaphase, they will all line up. Okay. During anaphase, they disjoin and the alleles segregate for each other. During telophase, now you have combination of chromosomes that is separated into different cells. So one cell you can have big A, small b, big A, small b. The other cell you have big A, small a, big B, small a, big B. And each one of the combination will become into different cells. Small a, small a, big B, big A, small b. Sex-linked gene in human beings. Because males need only to inherit one X chromosome to exhibit an X-linked trait, there are a preponderance of males who are afflicted with recessive sex-linked disorder. Some X-linked diseases are hemophilia, characterized by excessive bleeding because of lack of critical blood quality factor. Female usually focal blindness. They are caused by a faulty red green perception through red or green receptors in the retina. So here is hemophilia, an X-linked blood clotting disorder in the royal family of Europe. So as you can see in this family tree, a lot of the afflicted of the hemophilia, people who get the hemophilia, are usually males, as you can see from the square shape. Usually the males, the princess, will have the disease, but the woman would be just the carrier. They carry the gene, but they do not get the disease themselves. They would pass down the disease to their daughters and sons. Usually their sons will be afflicted with the hemophilia. So this is another one, color blindness. So as you can see, usually the male will get the color blindness, and the male who is afflicted with color blindness, he will pass down the gene to his daughters. His daughter will be a carrier. So in this picture, as you can see, the colorblind man in 2-4 makes his daughters all carriers in 3-2, 3-3, 3-4, and 3-2, 3-4 would marry somebody. Okay, and then what happens is that there will be a possibility of his son getting a disease, 100%. But for the daughters of 3-2, she will become a carrier. But the thing is, one of the daughters can also get the gene which are not affected with the color blindness. So he can get a half chance to be a carrier. For the son can actually have half chance to either be normal or become afflicted. So Fragile X syndrome can cause mental retardation. It is associated with a constriction in the long arm of the X chromosome. As you can see, the name of the disease is called Fragile X syndrome. As you can see, this chromosome have an overhanging part which are constricted, which easily breaks, so we call it fragile. This anomaly is caused by a long section of DNA containing repeats of short nucleotide sequence. Happens in 1 out of 2,000 men. So the human Y chromosome has some genes, 250, which is more than people expected. Several of the genes on the human Y chromosome, it is required for male fertility, and obviously a mutation in such a gene will interfere with a man's ability to reproduce. In comparison, it has identified more than 1,000 genes on the human X chromosome. So genes on both X and Y chromosomes are mostly near the ends of the short arms, and they are pseudo-autosomal genes, so they behave kind of like autosomes. Sex chromosomes and sex determination. In animal kingdom, sex is perhaps the most conspicuous phenotype, meaning that you can tell. Animals with distinct male and females are sexually dimorphic. Sometimes this dimorphism is established by environmental factors. In one species of turtles, for example, sex is determined by temperature. Eggs that has been incubated above 30 degrees Celsius hatch into females, whereas eggs that have been incubated at a lower temperature hatch into males. Discovery that human females are XX and human males are XY suggested that sex might be determined by the number of X chromosome or by the presence or absence of Y chromosome. As we now know, second hypothesis is correct. In human and other placental mammals, maleness is due to a dominant effect of the Y chromosome. The evidence of this fact comes from the study of individuals with an abnormal number of sex chromosomes. XO animals, they become females. XXY animals develop as males. The dominant effect of the Y chromosome is manifested early in development, where it directs the primordial gonads to develop into testes. Once the testes has formed, they secrete testosterone, a hormone that stimulates the development of male secondary sexual characteristics. So, you can see here, XX. In the absence of Y chromosome, no TDF is produced. But for XY, the SYRY gene is transcripted into TDF, testis determining factor. The TDF would cause the medulla of the gonads to become testes. Without the TDF, the cortex of the embryonic gonads will become ovary instead. So, with testosterone, with the testes, it produces testosterone, a hormone that initiates development of male sexual characteristics. So, the embryo will become a male because of the male sexual characteristics arising from that testosterone. With ovary, ovary does not have any testosterone, so the embryo will develop female characteristics. Researchers have shown that the TDF is a product of a gene called SRY, sex determining region of Y, which is located just outside the pseudo-autosomal region in the short arm of the Y chromosome. Discovery of SRY was made possible by the identification of unusual individuals whose sex was inconsistent with their chromosomal constitution, XX male and XY females. So, XX male, you can see that the chromosomes are XX, but they have SRY gene. So, despite being XX, they become male because of the gene. So, normal XY male, the SRY gene are on the X. The SRY gene are on the Y chromosome, not X. But for the XY female, despite having an XY chromosome, they are missing a segment that contains the SRY gene. So they grow up as a female. Testosterone is a hormone that binds to receptors in many kinds of cells. Once bound, the hormone receptor complex transmits a signal to the nucleus instructing the cell in how to differentiate. If the testosterone signaling system fails, these characteristics do not appear and the individual develops as a female. One reason for the failure is an inability to make the testosterone receptor. Testicular feminization syndrome, now more appropriately called the complete AIS, androgen insensitivity syndrome. This is a genetic disorder that makes XY fetuses insensitive or unresponsive to androgens, which is male hormones. Instead, they are born looking externally like normal girls. Internally, there is a short blunt pouch vagina and no uterus, fallopian tubes or ovaries, but there are testes in the abdomen of the inguinal canal. Here is the ochirectomy or removal of testes, which is undescended. For XY females, the testes are there inside of the body but not descended. They are inside, trapped inside, unable to be used. The gene for the syndrome is on the X chromosome in band XQ11Q12. The gene codes for the androgen receptor, also called the dihydrotestosterone receptor. The gene is mutant in the complete AIS. Because of the mutation, the cells cannot respond to androgen. If a woman has a mutation on one of her X chromosomes, the risk for each of her XY offspring to receive the gene have the syndrome is 1/2, 50%. In the picture, they are born with normal looking female genitals, just diagnosed only when menstruation does not occur. So this is the normal male with the wild type TFM gene. SRY creates testis determining factor, creates testes, which creates testosterone. TFM creates testosterone receptor, and the testosterone receptor complex signals a male differentiation. So testosterone must be received by testosterone receptor to make male secondary sexual characteristics. But with male with TFM mutation, testicular feminization, SRY creates testis determining factor, creates testes, creates testosterone, but the TFM mutation causes no testosterone to be made. So there is no testosterone receptor complex, no signal, meaning that the body will proceed to develop as a female. Sex determination in Drosophila. The Y chromosome in Drosophila, unlike the in a human, plays no role in sex determination. Instead, sex of the fly is determined by the ratio of X chromosomes to autosomes. Normal developed flies would have 3 pairs of autosomes, XX or XY, 3 pairs of autosomes, AA. A represents one haploid set of autosomes. Y chromosome is for male fertility. So, a ratio of X to A, 1.0 or greater will become female, 0.5 to 1.0 both sexes, but if it is 0.5 or less, then the fly will become a male. In other animals, so in Drosophila, in human beings, males produce 2 gametes, X bearing and Y bearing, so they are called the heterogametic sex. But in females in this species, females are the homogametic sex, meaning that for female humans, XX, for Drosophila, XX also. But in birds and butterflies, it is reversed. So males are the one that is homogametic, ZZ, but females are the heterogametic, ZW. So in birds, the male has ZZ, female has ZW. So when they cross together, you can have the female creating 2 gametes, Z and W, and the male only create one type of gamete, which is Z. The female egg will be created from the Z and W, but for the male, they need 2 Zs to become male. In haplodiploid system of sex determination, eggs are produced through meiosis of the queen. Sperm are produced through mitosis in the male. Hermaphrodites. Hermaphrodites have both male and female sex organs. Many species of fish are hermaphroditic. Some start out as one sex and then, in response of stimuli in the environment, switch to the other. For example, the clownfish, Nemo. Other species have both testes and ovaries at the same time but seldom fertilize themselves, like slugs, sea slugs. Like these reptiles and fishes whose sex is determined by incubation temperature, hermaphroditic fishes has no sex chromosomes. Dosage compensation of X-linked genes. Animal development is usually sensitive to an imbalance in the number of genes. Normally, each gene is present in 2 copies. Departures from this condition, either up to down, can cause abnormal phenotypes, sometimes even death. Females with 2 X chromosomes and males with only 1. In this species, how is the numerical difference of X-linked gene accommodated? Dosage. Three mechanisms compensate for this difference. For example, 1. Each X-linked gene could work twice as hard in males as it does in females. Okay, so because you have just one, so the X must work together doing the job of two X's. Number 2, one copy of each X-linked gene could be inactivated in females. In that one have to be inactivated in females to make it equal, because male only have one. Okay, female have two, so one have to be inactivated. 3. Each X-linked gene could work half as hard in females as it does in males, because you have two chromosomes, each will work 50%, the other will work 50%, now you have 100. Compared to males, they have only one X chromosome, that one X chromosome will work 100%. Extensive research has shown that all three mechanisms are utilized. The first in DROSOPHILA, second in mammals, and the third in nematode. In male DROSOPHILA, you have hyperactivation of X-linked genes. In DROSOPHILA, dosage compensation of X-linked genes is achieved by increase in activity of this gene in males. So, this is what you call hyperactivation. It involves a complex of different proteins that bind to many sites in the X chromosomes in males. So, it triggers a doubling of gene activity. When this protein complex does not bind in females, hyperactivation of X-linked gene does not occur. So, total X-linked gene activity in males and females is approximately equalized. In mammals, you have inactivation of X-linked genes. In placental mammals, dosage is achieved by the inactivation of one of the female's X chromosomes. The inactivation event occurs when the mouse embryo consists of a few thousand cells. At this time, each cell makes an independent decision to silence one of the X chromosomes, because they are two, they are female. The chromosome to be inactivated is chosen at random. Once chosen, however, it remains inactivated in all the descendants of that cell. Thus, female mammals are genetic mosaics containing two types of cell lineages. The maternally inherited X chromosome is inactivated in roughly half of these cells, and the other half, the paternally inherited X, is inactivated. A female that is heterozygous for an X-linked gene is therefore able to show two different phenotypes. For example, the Calico cat. In Calico cats, you have an allele for orange fur and allele for black fur. So they are heterozygotes. In the cell division as chromosome inactivation, some of the cells have their inactive X in the black allele, so it becomes orange. And in the other cells, the allele for orange is inactivated, leaving only the black allele to be activated. In that area will be black fur. An X chromosome that has been inactivated does not look or act like the other chromosomes. Chemical analysis show that its DNA is modified by the addition of numerous methyl groups. In addition, it condenses into a darkly staining structure called the bar body. This structure becomes attached to the inner surface of the nuclear membrane, where it replicates out of step with the other chromosomes in the cell. The inactivated X chromosome remains in this altered state in all somatic tissues. In germ tissues or your sex cells, it is reactivated because two copies of the same X-linked gene are needed for the successful completion of the oogenesis. So, as you can see, sperm and oocyte limit each other to become zygote. Okay, zygote will form XY or XX. If the zygote becomes XY, it becomes embryo, and then the cycle starts over. But if zygote is XX, okay, the embryo will form an X inactivation. So some of them will become somatic cells, some of them will become the germline. But during oogenesis, X is reactivated in oogenesis to create an oocyte, because you want all of your chromosomes to be accounted for. Serological studies have identified human beings with more than two X chromosomes. They are phenotypically normal females, apparently because all except one of the chromosomes are inactivated. Superfemale XXX is taller than normal but have learning disabilities. Often, all of the inactivated X's congeal to a single bar body. This observation suggests that a cell might have a limited amount of some factor to prevent X inactivation. Once the factor has been used to keep one X chromosome active, the others quietly succumb to the inactivation process. Okay, these are the sources. The end.